Hi, I'm Theresa Harbig, and today I will present to you OncoThreads, a tool for the visualization of longitudinal cancer molecular data. So cancer development is an evolutionary process, and even in cancers of the same type, the mutational patterns can be very different. And even in one patient, they can be very different. So one of the main goals of cancer research is to predict the influence of a treatment on a tumor. But first, we need to visualize the data. One of the visualizations to visualize cancer evolution in single patients is a fish blood. However, a fish blood is not scalable to visualize an entire cohort. We can visualize one patient at a time, and it is really nice to see the compositions of the tumors, but we need visualizations for entire patient cohorts to explore temporal patterns. One visualization tool for cohorts over time is EventFlow by Ben Schneiderman's group. And here, shared events are aligned in blocks. And this tool is focused on the sequence of events. So we want to see which events do the patients share and what is the sequence of these shared events. And it is not able to integrate multiple different data types for samples. So we cannot characterize a sample, for example, with a whole array of mutations. This leads us to the requirements for our tool. We wanted to develop a tool for the visualization of temporal cancer molecular data in patient cohorts. And it should be able to integrate multiple different data types, such as mutations, epigenetic data, gene expression data, or protein levels. Also, it should be web-based and compatible with the CBio portal, which you can see on the right here. It is a portal for cancer researchers where they can analyze and visualize their data. To come up with the requirements for our tool, we decided to do a design sprint, which is a technique developed by Google Ventures. In a design sprint, you want to come up with a tested prototype in only 5 days. And we met with our entire team, which consists of people from Novartis, people from Dana-Farber, and from Harvard Medical School. And we started the design sprint by identifying the requirements for our tool. So we created a map with all the potential steps that our tool might take. And on this map, we chose a target that we wanted to work on during the week. And on the next day, we started sketching competing solutions. So we all sketched the solution individually. And I will not go into detail about the solutions, but you can see that there's a lot of dots on there. There's small blue dots and a big orange dot. And this is the results from Wednesday, where we decided on the best sketch. So a blue dot means that one of us liked an aspect about the sketch, and the orange dot means that this is the winning sketch. So based on this sketch, we implemented a prototype on Thursday. And by implementing, I mean, we just created Apple Keynote slides. So there was no programming involved. Of course, since it was just slides, there's only one path through our tool that the users could take, and it didn't show them the entire functionality that we intended. On the last day, we then tested this prototype with potential users, and we collected and organized their feedback. And because the feedback was quite positive and we really liked the idea, we decided to implement our tool OncoThreads. And I now want to show you how OncoThreads works with a small data set by a study from Johnson et al. From 2014. And it contains 23 patients with glioma. For each sample, and each patient had samples of initial tumors and one or multiple recurrences. For each sample, we had overall mutation count, mutations, neoplasm histologic rate, and we also had information about the treatments that happened between the samples. So here I will show you how the tool works for one patient, but in the end, we will show you an entire cohort. So here you have one patient with 2 samples at 2 time points. The first time point is on the top and the second time point is on the bottom. And we have loaded 2 features, which are neoplasm histologic rate from 2 to 4 and mutation count. And in the first sample, you can see that the patient had a grade 2 tumor and a low mutation count of 39. And in the second sample, the cancer progressed to grade 4, and we had a very high mutation count. So the question now is there something that could explain this change? Or was this just the natural progression of the tumor? And maybe did the patient receive a treatment in between? In OncoThreads, we can add treatment blocks between time point blocks. And here we added a drug called temozolomide, which is a common anti-cancer drug. And as you can see, the patient received GMC in between the first and the second time point. And the question now is this a common pattern or is this just coincidence? So now we have to look at the entire cohort, which leads us to the first actual screenshot of OncoThreads. And now the patients are aligned, and it basically represents a heterogeneous heat map. And now we can just go through the patients and see if we can find similar patterns. And in fact, we find this pattern 5 times, but going through the patients is not a good solution. So how can we make this pattern easier to see? To facilitate finding patterns, we implemented 2 techniques called sorting and grouping. First, I will show you sorting. So what we want to do here is we want to sort the second time point by mutation count so that all the patients with a high mutation count move to the right. And after that, we will have some crossing edges. Therefore, we will realign the cohort based on this sorting. So now the entire visualization is sorted by mutation count in time point 2. And we can again see the pattern that we saw before, but it's now much more obvious. The other technique that I want to show you is grouping. So instead of looking at single patients, we now want to look at groups of patients. And more specifically, we want to look at patients with a high mutation count and a low mutation count. But mutation count is a continuous feature. And to group it, we need to bin it to make it ordinal, because with a continuous feature, we cannot create groups. And so I will first group the second time point by mutation count and bin mutation count, and then I will group the other time points. But you will see that in the video. So I bin mutation count into low and high. And this here is what we call a feature operation. And binning a continuous feature is only one example for that. We can do many other feature operations, such as combining features or transforming them into a binary feature, or using a log function to transform a continuous feature. But I will not have the time to show you all of that, so we will only see binning in this talk. So when I click apply, the time point will be grouped by mutation count. I'll group the other time points, and then I do some resorting. And then we can again see the pattern that we saw earlier as well. However, you might wonder now, why did we actually need to do this? Because we saw the same thing when we only sorted the data. And one reason for grouping is that it does scale better than sorting. So when we have a lot of patients, they might not fit on the visualization. But if we group the patients, the groups will always fit on the screen. Moreover, when we group the data, we can see the distributions of other features in the grouped feature. So here we can see the distribution of neoplasm histologic grade in mutation count high and in mutation count low as well. So we can see how many patients with a low mutation count had a grade 2 tumor, how many had a grade 3 tumor or a grade 4 tumor. And what we did by grouping is we iteratively transformed our heterogeneous heat map into a Sankey diagram. However, one thing stands out if we look at this visualization, and we could also already see it in the other visualizations. So There is TMC treatments here that are not in between the first and the second time point. But if we want to analyze the effect of TMC, we want all the TMC treatments to be in one block. So to do that, we implemented realigning the cohort. And for that, we can move patient columns. So far, samples are aligned based on the sample type, such as the primary sample, the first recurrence, and the second recurrence. But TMC treatment is not always between the primary sample and the first recurrence. So we need to align the samples based on the treatment. So what we want to do is we want to move this patient down, and these two patients we want to move up. And we can simply do this by selecting the patient and either select if we want to move him down or up. And now we could redo the analysis and see if the results changed. So far we added features because we had prior knowledge of the data and we knew which features might be interesting. However, sometimes it happens that we do not have that much prior knowledge and we still want to find features of interest. To do that, we first have to think about what are features of interest. And obviously, of interest depends heavily on the question asked. But there are some values that can characterize features to give you some information about patterns that you might find in the data. So for example, if we look at this cohort here, you can see that all the patients are the same. However, they all transition over time, but they always transition to the same value. So we can say that there's no variability between patients, but high variability over time. So by just looking at these two values, we can already know which kind of pattern to expect. Similarly, for this cohort, we have many different patients, but over time, each patient always stays the same. So we have high variability between patients, but no variability over time. And again, with these two values, we can already know that the pattern must look something like this. Probably one of the most interesting patterns is something like this, where we have some variability between patients and some variability over time. And if we find a pattern like this, we know that there might be subgroups that we can compare, which is always quite interesting. And therefore, again, with only the variability values, there's a way to characterize a feature. To measure or calculate the variability, we used standard statistical measures like the mod VR, the coefficient of unlikability, and the coefficient of variation. But of course, I will not go into detail about these measures. We then calculate these measures either for every patient or for every time point, depending on the kind of variability that we want to look at. So if we want to look at variability over time, we calculate these scores for every patient. And then we have one score for each patient. But because we want to have one score for each feature in the end, we need to aggregate these scores in a way. And we also do this by just standard functions like averaging the scores or getting the min or the max. And this again depends on what you want to look at. And we also want to transport that knowledge to the users. So we need to visualize it as well. So that's why we implemented something called our Feature Explorer. And this Feature Explorer uses a tool called Lineup, which was developed by Gratzel et al. And in this tool, tabular data can be ranked. So we basically have a table because we have the features as rows and as columns. We have attributes of these features, such as the data type or the range for continuous features or categories for categorical features. And we can also add our variability scores here. So we select a score for across-time variability and for within time point variability, and then we can rank the data, and we can always see which feature ends up on top or on the bottom, and therefore we gain some information about the features and their patterns. So as a summary, temporal cancer molecular data can be visualized using temporal heat maps and Sankey diagram. And we can rank our features based on their ability to identify patterns of interest. The design sprint technique helped us to develop a new concept within only 5 days, and it also helped us to create a shared vision for our team. Of course, OncoThreads is quite exploratory, so we decided to extend it with automatic clustering to identify disease states, which led to a new tool called thread states. And this is their teaser figure. There will be a talk about that today at 2:20 p.m. by Kuan-Yin Wang. And I very much recommend to you to go to that talk. And I thank you, and I'm open for any questions.